Is that fine? Yeah, I think that's fine. Okay, cool. Oh, wow. Okay, it's lots of people. Okay, so we'll get started today. Um, so just saying a quick hello to everyone, and then welcome to tonight's seminar. As you can see, it's a science seminar, and then I'll be taking you guys through it tonight. So my name is Kevin, and just a brief introduction of myself is, well, I graduated in 2019 from high school, so quite a few years ago. And then during that time, I achieved the highest ATAR of 99.95 and achieved around 4 state ranks in several subjects. So since then, in the last maybe 5 years or so, I completed a Bachelor's of Science at university, and then currently I'm just a medical student. All right. So, I mean, as many of you would know already, this seminar is brought to you as a joint effort by Dr. Jin Education and also Cogito Tutoring. Okay. So the current science program that they have offers tutoring for year 7 to 10 sciences. They also have HSC sciences for chemistry and physics. Now, I've been told that HSC biology is going to start next year. All right. In terms of the tutors teaching you guys for those subjects are all extremely high achievers, right? With either like a 99.95 ATAR or also being on the Australian team for the National Olympiad. So yeah, and just a little bit of housekeeping stuff before we get started. There's a lot of you and there's only one of me. So just make sure, everyone's muted so it doesn't disrupt the class. Okay, and then if you are able to turn on your video, that will also be great. Okay, just makes it a bit more personal and doesn't feel like I'm talking to a blank screen. All right. Now, if you have questions, there's going to be a questions time at the end of the seminar. So just hold on to them until possibly the end. All right. Cool. Now, as today's science seminar is going to talk about science reports and how to write science reports, I want to start with talking about the scientific method. All right. This may or may not be something you have heard of before, but I think it's quite relevant to the topic that we're going to talk about today. All right. So maybe you guys have heard about this, but the scientific method is essentially this logical approach to understand more about the world. Okay, because there's lots of things in the world that you don't really understand sometimes, right? And then you have to figure out why they occur and things like that. And the scientific method is just one of the ways we can, understand this world better. Okay, so there's quite a few things to talk about, but first of all, the scientific method is a systematic approach to gathering data to investigate a phenomena. Okay, so you're investigating a phenomena. Often you don't really know why that phenomena occurs, right? So you're a bit confused as to why it happens, but it's a method that helps you step by step figure it out. Okay, and what is the purpose of the scientific method? Well, the purpose is to formulate, to test, and or modify hypotheses based on observations and measurements. Okay, so we haven't really talked about it yet, and some of you may know this word hypothesis, but it's just this educated prediction on why it happens. Okay, but we'll talk a little bit more about that later on. Now, I want to move on to this next slide. This is the general structure of the scientific method. Okay, once I go through this, you may be like, oh, like I kind of already know this, but I still think it's useful to explicitly look at each individual step. Okay, I'll give you an example later on as well. All right, and it will make a bit more sense later, possibly. Now, first of all, you start with an observation, okay? As we go about our everyday lives, you make lots and lots of observations, all right? You look at the sun, it rises from the east. Why does it rise from the east, right? And that could be a question, right? It could lead to a question. So you ask a question, right? So like I said just then, why does the sun rise in the east, okay? Now, oftentimes you will have to do some background research, okay? Because if I'm asking about the sun and the direction it rises, I may need to know a little bit more about that before I can actually design an experiment and try to answer why that happens, okay? So after you have to do background research. Now, after you do your background research, you can construct a hypothesis. Like I said, that's an educated guess. educated prediction on why it happens. Okay? And after you get this hypothesis, you would design an experiment. Okay? And this experiment will test the hypothesis. Okay? So you test the hypothesis with an experiment. Okay? Now, during the experiment, you may find that the procedure may or may not work. Okay, maybe when you were designing the experiment, you forgot one or two important steps. And as a result, the experiment doesn't work. Okay, so depending on whether your procedure is working, there's either a no or yes. Okay, now if it's not working, obviously it can't go ahead, right? If it's not working, you have to troubleshoot it. Okay, you have to go and fix it back up, right? You have to add those one or two important steps. Okay, however, if you're lucky enough, right, on the first go, your procedure is working, right? And yes, it's working. What's going to happen is you're going to collect data, okay? You're going to analyze the data, and then you're going to draw conclusions, okay? Now, when you draw these conclusions, right, what we're trying to do is we're trying to compare it to our hypothesis, right? Remember, the hypothesis was an educated guess, right? Now, if your conclusion aligns with the hypothesis, so it agrees with the hypothesis, great, right? You can communicate your results and be like, yes, I did this experiment, all right? The results align with my hypothesis and it explains why this observation happens. Then that's great. However, oftentimes in science, right, you'll find that after you do the experiment, okay, and you draw the conclusion, it doesn't really agree with your hypothesis, and that's perfectly normal. Okay, and oftentimes in science, you do an experiment and it often fails. It doesn't really agree with your hypothesis, and that's perfectly okay as well. So look at this option. The results align partially or not at all with the hypothesis. In that case, since it doesn't agree with it, that's still fine. It's still very valuable. What you can do is you can use this failed result, okay, and go back and then try to design a new hypothesis and design a new experiment to test it. Okay, so regardless of whether your experiment is, a success or a failure, there is still always valuable lessons to be learned. Okay, now when we talk about communicate results, that's when the scientific report actually comes in hand, right? So you do all of this, okay, and then you're going to communicate the results, right? So you want to will convey your results and experiments to someone else. So you may have to write a scientific report. Okay. Now, before we talk about the scientific report, what I'm going to do is I'll give you an example. All right. Let's see the scientific method in action. All right. So look, everyday scenario, observation. I plugged in my phone to the charger and it doesn't charge. Okay. So that's my observation. My phone is not charging. All right. So the question that I'm going to ask myself now is, why is my phone not charging? Right. Hopefully you guys can see that's the first two steps. Right. Now, my hypothesis, so it's my prediction, right? My prediction is, well, the charging cable is damaged. Maybe it's the charging cable. All right. Maybe it's actually my phone. Maybe it's the power outlet. I don't know, but I have to make a guess first. My guess is it's the charging cable. All right. Now, I did my hypothesis. Now I have to test it with an experiment. Okay, so how do I test it? Well, since I used the same phone as my sister, and I know she charged her phone last night, what I'm going to do is I'm going to go steal her cable. I'm going to steal her charging cable and then plug it into my phone and then see if that works. Okay, now data collection and analysis. So it's all the way down here. All right, check if the charging symbol appears on my phone. All right. If it does, that means my phone's charging and perfect. It must be the cable's fault. But what if I plugged it in and it's not charging still? That means it's not the cable, right? So that's all part of data analysis and drawing conclusions. Okay. Now, like I said, my phone was not charging because the cable was faulty, because now that I use a new cable, the phone starts charging, right? So it aligns with your hypothesis and then you communicate the results. However, what happens if I got a new cable, it still doesn't charge? That means it does not align with the hypothesis. So what do I have to do? I have to design a new hypothesis and then test that now. Okay, maybe it's the power outlet that's broken, right? And then you come up with this whole other experiment to draw another conclusion, okay? But hopefully you guys can see this process is fairly useful, all right? You use it in your everyday life as well, okay? Not just in science. So it just helps you understand the world better, okay? It answers your questions about the world. Now, let's move on to writing science reports, okay? Now, why did I talk about the scientific method before the science report, all right? Well, the reason for that is, well, like I said, you have to communicate your results, all right? If you want to teach someone else or tell someone else what happened, you're going to have to communicate it. And the way of communicating with scientific data is using scientific reports, okay? Now, the scientific report, luckily, okay, mirrors, so it kind of copies the structure of the scientific method. And then once I show you the components of a report, you'll be like, oh, wow, it shares very similar steps and components, okay? Now, let's have a look at why a science report is important, okay? Now, it teaches you how to communicate clearly and accurately, okay? So writing a science report is quite a valuable skill, okay? So it doesn't matter whether, you're conducting an experiment for a project or whether you're trying to, I don't know, participate in a science fair or something like that. A well-written report can make a big, big difference in how your work is perceived and understood, okay? It has to be clear, okay? If it's not clear, even if it's the best report in the world, if nobody understands it, it's not very useful, okay? Now, why else is it important? It prepares you for higher education and professional careers, okay? So in terms of communication, right, it helps you, well, achieve better grades in school, of course, right? But communication is also something that you'll need throughout your whole life, okay? You have to communicate well with other people, okay? Now, you'll also develop several essential skills, such as critical thinking, data analysis, and maybe even attention to detail, okay? Things like that. So that's why a science report is quite important. It teaches you all these skills, all right? Now, these are my main objectives for the night, okay? So there's 4 things here. Number 1, understand the structure and the key components of a science report. So that's the main thing, actually, okay? The structure and key components. Number 2 is learn effective writing techniques to enhance clarity and conciseness, okay? So you don't want to be too wordy. You want to know how to write a science report that's, succinct, it's concise, and it's clear, okay? So hopefully I can give you some tips on that. And here, what else is there? Feel confident in your ability to write a high quality science report, okay? And lastly, this is a bit of additional stuff, okay? But begin to differentiate, okay, accuracy, reliability, and validity. These 3 things are quite important to the scientific field, okay? And it's often misunderstood a lot of the times. So, I mean, it's good to get you guys started early, okay? So hopefully we can have enough time to talk about this, all right? But the main chunk of it is the first 3 dot points, all right? Now, let's have a look. What is the purpose of the science report? Okay, so like I said before, the science report is a formal document that communicates, okay, its main purpose is to communicate the details of a scientific study or experiment, okay? So it helps convey whatever you did to someone else, all right? The primary purpose is to share your findings with others in a clear and concise manner, okay? And lastly, it allows other scientists, teachers, and peers to understand, replicate, or build upon your work. This last one is very important. Building upon your work, okay? Oftentimes when you do an experiment, okay, it may make a small advancement. It may not answer the whole question, okay? But what this science report allows it to do is, well, even though you made a small improvement, you write it down, you communicate it to someone else, they can use your work to make another small advancement, all right? And that's how, the scientific community can actually get more done, all right? You share knowledge and you build upon others' works, okay? So that's why the scientific report is very important. Now, key functions. There's 4 key functions I want you to understand. It's getting a bit repetitive now, right? But this is the last slide on this before we jump into it, okay? So learning, okay? So the key function, learning. Why, right? Well, the science report helps you develop, well, like I said, critical thinking, data analysis, and even writing skills, right? And a whole lot other more skills as well, okay? Documentation. So they provide a detailed record for your research process and results, right? Sometimes you will have to refer back to old experiments, right? And that's why it's useful to have documentation, right? Because you'll forget what happened and whatnot. So it's useful to keep a record. Communication, I've already talked about this a lot, okay? You're trying to teach and communicate with other people, okay? And lastly, validation. Remember this whole thing about building upon your work, okay? Sometimes you're doing an experiment and you think you made an advancement or, you know, you discovered something important, but maybe you made a small mistake in your experiment. And someone else may come along, do the same experiment and figure out, well, they didn't get the same results. Right? So in that case, that means your experiment may not be valid. All right. So it also helps other people validate your experiment. All right. So 4 key functions. Now, let's jump into it. The structure of a science report. It looks quite a lot, but it really isn't. Okay. A lot of this is quite simple. Okay. So there's 10 things, right? A nice number. Okay. Starting with the title, abstract, introduction, materials, risk assessment, method, results, discussion, conclusion, and references. We'll go through each one, okay? And I'll give you examples of good and bad ones, all right? So hopefully you guys can actually learn something and then maybe carry that forward in your studies, okay? Now, in order to actually give you those examples, I have to come up with an imaginary scenario or imaginary experiment. So this is the experiment of the day. Maybe not day, but you know, night, okay, of tonight. This is the experiment that we're going to focus on, right? So investigate how different light conditions will affect bean plant growth, right? So obviously you guys know, in order for plants to grow, they need water, they need sunlight and whatnot. But here I'm investigating, you know, how different light conditions will affect bean plant growth. Okay, so when I talk about different light conditions, the different light conditions are here. There's 3 of them. Number 1 is just normal sunlight. Okay, that's just like you put the plant in normal sunlight and then see how it grows. Number 2 is artificial light, right? I'm sure lots of you are using either a light or like a table light or something like that. That's artificial light. Okay, so it's not natural sunlight, but it's also a light source. Okay, and number 3 is darkness. Okay, so I drew a little diagram for the setup as well. Okay, so in sunlight, I drew a little sun, and then you can see 4 bean plants. Okay, there's artificial light, so you can see there's a lamp. Okay, and then there's also 4 bean plants. There's a box around it to make sure it's not getting any natural light. Okay, and darkness. Well, darkness, you have a box around it, and you have 4 bean plants, and it doesn't get any light at all. Okay, hopefully looking at this, you guys can already make, you know, some estimations as to which one's going to grow the most. Okay, well, obviously it's going to be the sunlight one because that's the most natural one. Okay, darkness, the plants in darkness probably won't grow that well because it doesn't have sunlight. Okay, now the question is, what about artificial light? Right, that's the thing, right? Is it going to be as good as sunlight? Okay, is it going to be better than darkness? Right, we don't know. So we're going to investigate. Okay, there we go. Now, this is the experiment. Let's go forward and look at the different components. Okay, title. This is number 1. So on top here, you can see a timeline. Okay, so we're on the very first one, the title. The purpose of the title is to provide a concise, okay, and descriptive summary of the report's content. Try to keep it as just 1 sentence. You don't have to write multiple sentences. It's a title, okay? Now, what are some tips? Keep it short, see? Keep it short. Keep it specific and relevant, all right? You don't have many words for your title, so you want to choose the best words to use, okay? Now, try to give the reader a clear idea of the purpose of the experiment and the variables being studied. Okay, so I'll talk a bit about the variables later on, but if I go back up here, you would see the variables, okay, in this case, it's going to be the different light conditions, okay, and the bean plant growth, okay? Now, have a look at this example. The effect of sunlight, artificial light, and darkness on plant growth and how they are different. Okay, now have a think. I'll give you 10 seconds to think. Is this good or is this bad? Have a think. 10 seconds. Okay. Well, the answer here is actually it's not a great title. I'm going to label this as a bad title example. This is a bad example. Okay? Why is this a bad example? First of all, it's too long. First of all, it's too long. It's too wordy. You're using a lot of words. Remember, we'll keep it short, keep it specific and relevant. Okay? Now, what else is there? Well, I would say it contains redundant. It contains redundant phrases. Okay, what do I mean by that? Well, I would actually think this last bit here is a bit redundant. It's a bit unnecessary. It seems just like a repeat of what was said before. Okay, why? Because when you talk about the effect of sunlight, artificial light, and darkness, right? You're already aware that it's different light conditions. Okay, so it's already implied that they are different. Okay, so you don't actually need this last bit. I would say it's redundant. It's a bit useless. It doesn't add any more value. Okay, so this is a bad example. Okay, now look at this one. Plants and light. Is this a good title? I don't think so, right? First of all, it's got 3 words, plants and light. How do I know what this report is going to be about? Right? So the previous one was too long. Okay, and it's redundant. But this one, first of all, it's too short. Okay, it's too short. And because it's too short, it becomes too vague. You actually do not convey any information whatsoever. Okay, so it literally tells you no information about the experiment. Tells you no information about the experiment. Okay, I don't even know what this science report's going to be on about. Okay, if I read this title. So once again, bad example. Okay, don't do this. Now let's have a look at this. Okay, the impact of different light conditions on bean plant growth. Okay, so this one is pretty decent. Okay, first of all, it's clear. Okay, it's clear and specific. Okay, it doesn't use too many words. Okay, what else? It actually tells me what plant I'm using, the bean plant. Okay, now this is the reason I chose the bean plant. And that's because, well, when you conduct an experiment, you have to make sure that you're able to finish that experiment, right? So I don't want to design an experiment that takes 50 years to finish. That's too long, right? Now, this experiment is designed for maybe 2 weeks or 3 weeks or something like that. All right, I actually used 2 weeks, and then not many plants actually grow in 2 weeks in order for you to measure the growth. Okay, but the bean plant grows very quickly. Okay, it's very easy to handle. It's quite sturdy as well. It doesn't die easily, so that's why it's pretty good. Okay, so it actually states the plant, so it specifies type of plant. Okay, specifies the type of plant, and it also indicates the variables. Indicates variables. The variables being, well, first of all, light conditions, and second of all, the bean plant growth. Okay, those are your variables. Okay, so this is actually a good example of a title. All right. Now, let's move on to this thing called the abstract. Now, the abstract may actually be optional for high school students. Okay, so in the real world, whenever we write a science report, we use an abstract. It's optional for you guys because you're a lot younger. Okay, now the abstract, right? What's the purpose? The purpose is Well, to offer a brief summary of the report, highlighting the aim, the method, the results, and conclusions. Well, you can see that's pretty much a lot of the things you're doing in your experiment, right? So the purpose of an abstract is to act as a brief summary. Whenever you're doing science research and whatnot, right, science papers can get quite long, okay? You don't have the time to read the entire paper and read 20 of those, okay? So often we'll read the abstract. The abstract tells you the key ideas, the aim, the method, the results, and the conclusions. Now, if I read the abstract and I'm like, oh, that sounds like it's gonna be helpful, I will go and read the whole paper. But sometimes if I read it and I'm like, oh, it's not very helpful, then I won't read the paper. It saves me a bit of time. Okay, now, because the abstract talks about aim, method, results, and conclusions, you can see, well, conclusions here, the results here, right? Method is here. It uses a lot of the things from later on. You can see it uses stuff from later on, right? So the tip here is you write this last, write it last. There's no way you can write it at the beginning because you don't have your method nor conclusions, right? So it's quite short. You keep it to maybe 150 to 250 words, okay? And ensure that it covers all the key aspects of the report, okay? Now, this is an example of an abstract. Let's have a quick read, okay? Now, this experiment was about growing bean plants in different types of light. We put some plants in sunlight, some under a lamp, and some in the dark. We watered them every day and measured how tall they got. The plants in the sunlight grew the most. The ones under the lamp grew a little, and the ones in the dark didn't grow much. We wrote down the measurements and looked at the plants to see what happened. This showed that light is important for plant growth. Now, is this a good or bad abstract? Have a think. I'll give you guys 10 seconds again. Okay, the answer. Oh, someone's got the hand up. Okay. I can't really see the hand up anymore. Izzy, you can feel free to unmute and tell me real quick, do you think it's good or bad?
I think that it's good.
You think it's pretty good? Okay. Why do you think it's good?
Because it says what they did and the result of what happened.
Okay. Result, what happened? Okay, that's a pretty good answer, Izzy. So thank you for that. Now, what I'm going to do is, before I tell you whether this is good or bad, read this next one. I prepared another one. It's a bit longer, but it's not too long. So have a read of this one. This experiment aimed. Oh, it aimed. So it tells you the aim. To investigate the effect of different light conditions on the growth of bean plants. The study was conducted using 3 groups of plants exposed to natural sunlight, artificial light, and complete darkness, respectively. Over a 2-week period, the growth of the plants was measured and recorded weekly. The results showed that the bean plants exposed to natural sunlight exhibited the most significant growth with an average height of 14.6 centimeters, followed by those under artificial light, which has an average height of 11.6. Plants kept in the darkness showed minimal growth, averaging 4 centimeters in height, and displayed poor or signs of poor health, such as yellowing leaves and weak stems. These findings highlight the crucial role of light in promoting healthy plant growth and development, with natural sunlight being the most effective light source for bean plants. So compare this one and the one before. So I'll go with Izzy again. So have a think again, Izzy. What do you think about this new one?
I think this one is better than the last one because it's more detailed.
Yes, correct. Right, thank you, Izzy. So quite a few of you, but sorry, I could only pick one for the sake of time. But thanks, Izzy, for that. You are correct. This one is more detailed, and hopefully you can see why it's more detailed. First of all, look, it tells you it's 2 weeks. It's 2 weeks. This previous one didn't tell you how long it is. All right? Did they use any numbers at all? No, it didn't tell you the average growths and stuff like that as well, right? So here, there's average growths. It uses data to support it as well, okay? So if we go back to here, remember an abstract is a brief summary of the aim, method, results, and conclusions, right? This one also has an aim, right? It told you the aim. It's very clear, right? So I'm going to go back up here and call this a bad example, okay? It's a bad example because why? It lacks detail. It lacks detail, okay? There's no specific findings. It doesn't use any numbers to support it, okay? It doesn't convey the results very well, okay? And one thing I'm going to say as well, it's informal language use. Informal language that's being used. Okay, so whenever you write a science experiment, try to avoid saying we, like I did this, we did this, blah, 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 right? So instead of saying we and I, you want to try to use a passive voice. So a passive voice brings the attention away from the actual people, okay? But it rather instead focuses on the process. So instead of saying we measured the temperature, right? You can say the temperature was measured, right? So use the passive voice, okay? Now, this one is a good example. As you guys can see, it's very detailed, okay? It's not overly long, and it gives you a good idea of what this experiment is going to be about, okay? So I'll read this abstract, and I'll be like, oh, that sounds pretty helpful. I'm going to read the rest of the paper. Okay, so after the abstract, we have the introduction. Okay, so here we have the introduction. What's the purpose? It provides context for your experiment. Okay, it provides context. Why are you doing this experiment? Okay, why is it important? Okay, explaining background information and relevant scientific concepts. Okay, now two important things in the introduction, okay, is the aim and the hypothesis, or otherwise the research question, okay? Now, in terms of the tips, start broad, okay? Start broad and then narrow it down to your specific topic. Clearly state your aim. So we need to clearly state the aim and hypothesis. These two are very important, okay? So let's have a look first at what is an aim. Okay, so the aim is a statement outlining what the experiment is trying to validate or investigate. Okay, and the aim should contain both independent and dependent variables. All right, so we talked a little bit about variables, but I haven't told you guys independent and dependent variables. Okay, so real quick, although I'll do it again later on, the independent variable is the variable that you will change. It's the variable that you will change. And if you have to think about it, what am I changing in this experiment? I am changing the light conditions. That's what I'm changing. So that's the independent variable. The dependent variable is different. Okay, it's called dependent because it depends on the independent variable. Okay, so what's going to happen when you change the light conditions? Well, when you change light conditions, most likely the plant growth is going to change. So in this case, the plant growth is the dependent variable. Okay, so in the chat, I see some people talking about the slides. That's okay. At the end, I'll try upload the slide here, and then you guys can have a copy. So just have a listen for now. Okay, don't worry too much about missing the stuff before. Okay, now we talked about the aim. Let's talk about the hypothesis. I've told you guys already, but the hypothesis is a prediction based on evidence and prior knowledge or observations. Okay, so it's based on evidence and prior knowledge. Okay, so do you guys remember at the very, very beginning, we talked about the scientific method? Where was it? Here. The scientific method has a little bit on doing background research. Why do you need background research? Because when you construct a hypothesis that's based on prior knowledge. Okay, it's based on prior knowledge. That's why you need that background research. Okay, so I'm going to come back down to here. Where was I up to? Hypothesis. Yeah, okay. The hypothesis must always be testable. Okay, it must be testable with an experiment. And it must clearly state the independent and dependent variables. All right, so similar to the aim, but this time I'm making a prediction. All right, cool. Let's have a look at this one. So give this a read. Plants need light to grow. Without light, they can't perform photosynthesis, which is how they make food. We wanted to see how different types of light affect plant growth. We used bean plants for this experiment. We put some plants in the sun, some under a lamp, and some in the dark. We watered them every day and measured how tall they got. We think the plants in the sun will grow the most. This experiment is important because it shows how light affects plant growth. Okay, now let's choose someone new. Derek, what do you think about this one?
Okay, so this one is not the best one, I'd say, because it uses informal language. It says we used, we put, we think.
Yep. Okay, that's pretty good.
And something to add for we think. It's from their opinion that we think they need to fully prove what has happened.
Okay, yeah. So when they say we think, right? Often when you make like a prediction or stuff like that, you have to back it up with something. Okay, you have to back it up with some background research, right? So pretty good, Derek. Nice. Okay, so I'm gonna just mute you a little bit for now, and then let's have a look at this next one. Okay, so this is another example of an introduction. Now, right off the bat, what do you notice? The structure is more clearly laid out, right? Can you guys see there's the background information? It has an aim and it has a hypothesis, right? It clearly states each of these things. But when you look back here, you're like, oh, where's the aim? Right? You'll be reading and you'll be like, where is the aim? Where is the hypothesis? Okay, where's the hypothesis? Right? So the hypothesis was actually supposed to be this one. All right, but as you can see, it's not very clear because it's just one big paragraph, right? You don't structure it properly. So don't be afraid to use paragraphing, okay, like this. Okay, so remember, with a good introduction, you should start broad. So you're starting broad with the background information, right? Light is a crucial factor influencing plant growth and development. It plays a fundamental role in photosynthesis, the process by which plants convert light energy into chemical energy to fuel their growth, right? Different light conditions can significantly impact various aspects of plant physiology, including growth rate, leaf development, and overall health. Understanding how plants respond to different light environments is essential for optimizing agricultural practices and indoor gardening. So can you see, even towards the end, it tells you the significance. This is the significance. Okay, it tells you why it's important. All right, now you have the aim over here. All right, the aim of this experiment is to investigate the effect of different light conditions, natural sunlight, artificial light, and complete darkness on the growth of bean plants. So like you guys can see, the different light conditions and the growth of bean plants, those are your variables, right? Remember, when you talk about the aim, you have to include your variables, okay? By comparing these light environments, we seek to determine which condition promotes the most robust plant growth. So that's the aim. What's the hypothesis? What is your prediction? Well, you can simply start with, it is hypothesized that bean plants exposed to natural light will exhibit the most significant growth compared to those under artificial light or in complete darkness, right? Now you have to back it up because you made a hypothesis. Where is your evidence or prior knowledge? This hypothesis is based on the assumption that natural sunlight provides a full spectrum of light, which is most beneficial for photosynthesis and overall plant health. Conversely, plants grown in darkness are expected to show minimal growth and signs of poor health due to the lack of light necessary for photosynthesis. Okay, and once again, we're finishing with why it's, well, what you're trying to do here, the purpose, right? By conducting this experiment, we aim to highlight the importance of light quality and availability in plant growth and provide insights into the optimal conditions for cultivating healthy plants. So it's much better, okay? It first of all sounds more formal, okay? It sounds more academic because we're not using we, and I did this, I did that, right? You're using a passive voice, okay? So this is a good introduction, right? And it's good because, well, it does everything well. It talks about significance, aim, hypothesis, and stuff like that, okay? Now, let's move on from introduction to materials. So we're up to number 4 now, okay? Let's look at materials. This one's pretty easy. Purpose, list all the equipment and resources used in the experiment, okay? Ensure that the experiment can be replicated, okay? Can be replicated by others, okay? Now, tips, provide a complete and detailed list. So it must be complete and it must be detailed, all right? Someone else should be able to gather this material list, okay? Gather all the materials and perform the same experiment. If they can't do that, then that means your materials list is lacking, okay? So specify quantities and sizes where applicable, okay? So quantities and sizes. Mention any specific brands or models if they are relevant, okay? Mention any specific brands or models only if it's relevant, all right? Now, let's have a look at this example, a materials list, right? Have a think, good or bad? Okay, I'm looking at this, right? And it doesn't seem that good, right? Jacob, someone new. Jacob, what do you think?
I think it's bad because it doesn't specify like the amount of soil or how many bean seeds.
Correct. Very good, right? It doesn't tell you the quantity and it's not even complete, right? Remember, we have to use artificial light, right? Where is the artificial light? Where is the lamp, right? You don't see anything here. Again, bean seeds, how many bean seeds, right? So it's not very good. So this is a bad example, okay? It doesn't tell you quantity, okay? It's not complete, okay? So it's not good. Look at this one. This one's better, okay? But have a look at why it's better. 12 bean seeds, 12 small pots with drainage holes, 12 labels, 5 kilos of potting soil, gloves, the lamp with a 12-hour timer, cardboard boxes. Why is that needed? Well, to actually create the darkness environment. And even for the artificial light, I'm putting a box over it. So you need to specify the boxes. And look, I even put dimensions, right? 1 meter by 1 meter by half a meter. And then look, 250-meter measuring cup, because we have to give them water. If we don't give them water, they're gonna die, right? And you're gonna have to measure the plant growth. So you're gonna use a ruler, right? It's a 30-centimeter ruler. And then you're also gonna have a notebook and pen, right? So hopefully you guys can see this. It's much better. This is a good example. It's a good example, okay? Now, that's that for your materials. We're up to number 5 now, right? We're slowly moving along. Risk assessment. Now, some of you may be a bit young to talk about risk assessment, but eventually I would say it's quite useful, okay? And it is part of a scientific report, okay? Now, the purpose of risk assessment is to identify potential hazards associated with the experiment and outline measures to mitigate these risks. Ensures safety of everyone involved in the experiment, okay? Safety. Whenever you're conducting an experiment, you have to ensure safety, right? There's no point doing an experiment if someone's going to get injured, okay? Or if someone's going to get, hospitalized. That's no good. You don't want to do that. So you have to perform risk assessment. When you think about risk assessment, think about what are the possible hazards and risks to you, okay? To other people, to the environment, okay? You must think about how likely is it that there will be an injury or damage, all right? And if there is an injury or damage, how serious are the consequences, okay? Now, when you think about those, right, you can actually use a table like this. This is called a risk matrix. Okay, this is what you use to determine whether this experiment should go ahead or you shouldn't go ahead. Okay, so how do you read this? Across the top, you have consequences. Okay, so this way is consequences. This is your consequences. All right, and the going down is your likelihood. So downwards is likelihood. All right, now the consequences can be negligible, marginal, severe, or catastrophic. Okay, so catastrophic is kind of like someone is going to break a bone or someone might die from the experiment. Okay, so that's not good. Negligible means, well, it doesn't really mean anything. Maybe your skin gets a bit red. Okay, maybe you get a tiny cut if something happens. Okay, now in terms of likelihood, is it rare, unlikely, possible, likely, or certain? Okay, and then what you're going to do is, for example, if something is catastrophic, so someone might get hospitalized, okay, and it is certain to happen, okay, that's not good. There's an extreme risk, okay, you don't want to do this experiment, that's not good, okay. Now, what we're trying to do is we're trying to keep it at a low risk. I want to keep it to a low risk. I like it when it says low risk. So these ones are good. Okay, especially for the extreme risk, do not do it. Okay, if it shows extreme risk, you have to design a new experiment. Don't go ahead with it. It's dangerous. All right, so that's your risk matrix. Now, this is an example of a risk assessment table. Okay, so this is possibly what you put in your report. Okay, so this is an example. All right, so look, it tells you the hazard, the potential risks of the hazard, and how to control it if anything happens. Okay, so these are very important. Make sure you try to include these three columns. Okay, so one of an example of a hazard, it's not relevant to our example, but it's an acid. Okay, hydrochloric acid. So acids are not great. Okay, why? Because it's corrosive to the skin and to the clothes. Okay, so that's a potential risk. It can get on your skin, it can get on your clothes, and it's corrosive. All right, now control measures. Well, how are you going to deal with it? Well, clean up any spills immediately. Okay, when you're using these acids, wear safety glasses and gloves. Okay, when you're handling the chemical. All right, so that's a control measure. So that's an example. Another example is glass beakers. All right, in the science lab, you have a lot of things made of glass. Okay, and the thing with glass is you drop it, it's going to shatter, it'll break. Right, they can smash easily if dropped. Okay, the broken glass can cause cuts and lacerations. So obviously, if you drop something, don't pick it up with your fingers. It may cut you. Okay, so what's the control measure? You handle glass beakers with care using both hands. Right, you keep glass beakers away from the edge of the table. So keep it towards the center. All right, if the glass beaker breaks, immediately notify the teacher, okay, or an adult. Do not try to pick up the broken glass pieces. Okay, so as you can see, it's pretty logical. The hazard, what may happen with the hazard, and how to deal with it. Okay, it's quite logical in that sense. All right, now scroll down. This is an example of a risk assessment table for our experiment. Okay, so our experiment is not very dangerous at all. Okay, there's nothing crazy. So what's a hazard? Handling the plant. Okay, potential risks: skin irritation, minor cuts. Okay, control measures: wear gloves when handling the plant. There you go. Right, it's not too bad. That's probably low risk. Okay, handling water. Okay, potential risk: you can slip from water spills. Right, that's possible. So how do you control that? Well, you wipe up any spills immediately. Okay, you use a watering can, okay, with a spout to prevent spillage, possibly. Okay, that's one way to control it. What's another hazard? Artificial light setup. Okay, so remember I'm using a lamp. Now, what's the potential risk? Electrical hazards or burns. Because it's an electrical lamp, okay, you can have electrical hazards or burns. So what's the control? Well, ensure all electrical equipment is in good condition and you keep liquids away from electrical sources. So don't keep water near it. Okay, so it's pretty simple. All of these low risk. So therefore, I can go ahead with my experiment. I don't have to change much. All right? Cool. Method now. So let's talk about the method. Now, the purpose of the method, you guys will know, provide a detailed description of the procedure and techniques used in the experiment so that others can replicate the study. So once again, replication is key. You must contain enough information so that someone else will read it and they can replicate it. Okay? Now, the tips. Try to start the sentence with a verb. Try to start with verbs. Be concise, clear, and thorough. All right? You don't want to use too many words. Use a passive voice, okay? Don't say, I did this, I watered the plants, okay? Say the plants were watered, all right? Try to write in the past tense, okay? It's actually arguable. Some people like to write in the present tense. That's fine, honestly. It's just traditionally we write methods in the past tense because by the time you're writing the report, you already did the method, right? The method is already done. That's why you write in past tense most of the time. But regardless, whether it's past tense or present tense, keep it consistent. If it's consistent, I think you're fine. All right. And then a lot of the times, including diagrams. Okay. So remember, a picture is worth 1,000 words sometimes. Okay. And so is a diagram. All right. Now, example, look at this, a method. Put plants in different light conditions. You watered the plants every day, measured the plant's height sometimes, wrote down what happened to the plants. Tommy, do you want to tell me good or bad?
I think it's really bad because it never really said it says different light conditions, but it never said which light conditions.
Good.
It said it's also not consistent because it says put in the first one, but now it says watered the plants every day. So it doesn't really have like the same tense.
Okay. Put plants in different light conditions. That could be arguable. That first one could still be past tense, but I get where you're coming from, right? You're trying to keep the tense consistent. Yep. And is there anything else you want to add?
I think it's just maybe not like maybe it's just not specific enough, maybe.
Yes, not specific enough. Not specific enough. Okay, it's quite vague. All right. Now, let's have a look at this example. Okay, let's have a look. You labeled 4 pots for each light condition: sunlight, artificial light, and darkness. Okay, you filled each pot with potting soil. You planted 1 bean seed in each pot 2 centimeters deep in the soil. You placed sunlight pots on the windowsill where they received natural sunlight. You placed artificial light pots under a cardboard box with the LED lamp that has a 12-hour timer. So this lamp will turn on every 12 hours to provide light. Place the darkness pots under the second cardboard box to remain completely in darkness. We're not done. Oh, it crashed. Okay, sorry guys, give me a second. It just crashed on me, but hopefully, yeah, there we go. Okay. Number 7, you watered each pot with 50 mL of water every day. You measured the height of each plant at the start of the experiment using a ruler. Then you measured and recorded the height of each plant once a week for 2 weeks. Okay. And you used a notebook to record weekly height measurements and general observations of the plants. And I put a little diagram here. It's not the best, but I mean, I just used the same one as before. Okay. Grace, you want to tell me why this one's a bit better than the last one?
It's a bit more specific.
Yes.
Well, no, it's way more specific.
Way more specific.
Yeah, it's like 2 centimeters deep in the soil.
Good. Yeah.
It's just really specific.
Mhm.
Because whereas the first example, you can't really get any information from it.
Okay.
It's just 4 different.
Yeah, it's just 4 tiny steps, right? It's quite vague. It doesn't actually have any numerical values as well. See, I said label 4 pots. okay, 2 centimeters deep. And even watering the plants, right? You water with 50 mils of water every day, okay? So it's quite specific. It tells you exactly what you need to do, okay? And look at all the tense. The tense is consistent. It's past tense. I use the passive voice, right? So all of those things, right, is done in this method. So it's a good example, all right? Cool. Let's have a look at the results now. Results, purpose, obviously to present the data collected during the experiment in an organized and clear manner. Okay, what are some tips? Well, when we talk about results and think about results, we think about tables, graphs, charts, okay? Whatever medium is appropriate to display your data, okay? Now, the one thing that students do in results is they often try to analyze the result and try to interpret the result, okay, in this section, but that doesn't belong in this section, okay? The actual analysis of results actually happens in the discussion section. Here, all you're saying is, here is my data, that's it. In the discussion section, you'll be like, this data happened because of this, blah, 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 whatever, okay? But don't interpret your results in this section, okay? So, I mean, here, I'm just showing you, there's lots of charts, lots of graphs, you know, pie charts, scatter graphs, bar charts, and whatnot. There's lots of ways to convey your data, but you have to think about what's the most appropriate way, okay? We don't have enough time today to talk about every single one, so, I mean, I'm just gonna show you some examples, all right? Now, look at this example, all right? So, I mean, I've only talked about the sunlight plants here, so sunlight plant 1, 2, 3, 4, okay? I measured them on day 1, sorry, or day 0, okay? Measured it 1 week later and then 2 weeks later. So as you can see, well, it is growing, okay? It is growing. So that's a good thing, at least. In sunlight, the plant is growing. Very good, okay? Now, here's some observations. It says the plants look healthy. Okay, great. Is this a good example? Is this a good example? Izzy, back to Izzy, I guess. Not too sure why it keeps crashing. Maybe Bella? Bella, do you want to give this a go?
Like the observation, like when it says the plants looked healthy, it's not really giving any other like it's just saying that it looked healthy. It's not saying that, oh, like this plant grew a little bit more because of this plant grew more.
Yeah.
It's not saying why the sunlight affects like the plants like really well.
Yeah, okay. So, yeah, you're right. The plants look healthy. It's not great, right? It should be a bit more specific than that, right? Are the leaves green? Okay, are the stems strong? Okay, stuff like that. But, you know, when you just say it looks healthy, that's not great. Okay, it's not good enough. Now, what I'm gonna also show you guys the next one first, and then we're gonna talk about it. Look at this example, all right? Similar way of presenting the data, but look, okay? 2.0, 8.0, 15.0, okay? And then, I mean, all the other numbers are the same, just with a decimal point after, okay? The growth, all right? The leaf color and the stem strength, these are all observations they made. So you can see it's more specific, okay? And they also commented on the overall appearance, all right? So, I mean, Lucas, do you want to tell me why this one's better?
This one actually has a unit and it's more specific inside everything else. So it's the observations like the growth, the leaf color, and the stem strength and overall appearance. Because in the previous one, it's not very specific.
Yes. First of all, Lucas made a very good point. See, I deliberately left out the units. Is it 2 centimeters or meters? Right? We don't know. Okay. So the units are not here. Right? Make sure whenever you draw a graph, you know, make a table or anything, you have your units. Okay. Another thing is there's decimal places. Right? So whenever you use a 30 centimeter ruler, right, you can see there's intervals in between because there's millimeters. Right? So look at this. This one over here, it says, well, I mean, it says 2 for this, but for this one, it says 14.5. Okay, even though it is exactly 2, you should say 2.0. You should keep it consistent in terms of the decimal point, right? It's a very small thing, but I mean, it looks much better when you keep it consistent, right? And observations, obviously, there's growth, leaf color, stem strength. It's much more detailed, okay? And even on this side, there's a mean height, okay? It calculates the mean height, which is the average height of all 4 plants, okay? So that's pretty good in terms of an addition as well, okay? The mean height. Now, this is a good example of result, okay? Now, there's other ways of displaying your results, okay? Maybe in terms of a graph like this, okay? Have a look at this. So the yellow one is sunlight, the orangish one is artificial light, and then the red one is darkness. So as you can see, darkness grew the least overall. Okay, so it went from here to here, that's it. Okay, sunlight grew the most. It went the most furthest up. Okay, and then artificial light is somewhere in the middle over here, right? So a graph like this displays all that information, okay, in one place, which is pretty nice as well. Okay, now there's a lot more things to talk about when actually drawing a proper graph. This is not a perfect graph, okay, but it's just that I don't have time to talk about all the details of a little graph here. Okay, now another example is a table like this. Okay, so comparative table. So light conditions here, you have sunlight, artificial light, darkness. You have the mean height, so the average height that they grew to. Okay, the leaf color, stem strength, overall appearance. So when you combine them all into one table, you can compare them. It helps you compare better. All right, so that's another example. Now let's go to the conclusion, or not conclusion, discussion, number 8. We're nearly there, okay? What is the purpose of the discussion? So remember, I told you in the results, you're just showing the results, okay? You don't actually analyze the results. You don't have to say, well, the plants in darkness grew the least and the plants in sunlight grew the most. That belongs in the discussion, okay? So here is where you interpret the results, okay? Please, please, please do it here. Explain the significance, okay? Relate the findings to the original hypothesis, okay? You can assess the validity, okay? The accuracy and reliability. This one I'll talk about later, okay? Because right now it may not make a lot of sense. So I'll talk about this later, but it belongs in the discussion. Suggest possible improvements for future experiments. Now, this is a big thing, okay? You should reflect on your experiments and your results, okay? And think about what you can improve for future references, okay? Because someone may pick up your science report and be like, oh, he suggested some improvements. I can go and try and make these improvements and see what happens, okay? Now, what are some tips? Discuss any patterns, trends, anomalies. Consider possible explanations and implications of your findings, all right? Now, once again, have a look. Example of a discussion. All right, let's see whether this is good or not. The plants in the sunlight grew the best, which was expected. Okay, why is it expected? Right? The ones under the lamp grew too much. The ones under the lamp grew too, but not as much. The plants in the dark didn't grow much at all. This shows that plants need light to grow. The experiment went well, and we saw that light is important. Next time, we could try using different plants or different lights to see what happens. This experiment proves that light is necessary for plants. Okay, very simplistic. All right, missing a lot of details as well. All right, Izzy, do you want to share what you think about this? Have your hand up.
Like, it's really not detailed, and it needs to be like how much it grew, like how long, like how tall was it, and it doesn't give us a lot of information, so it's not good.
Yes, correct, right? So we actually have results, we have data. It doesn't mention anything about the data, right? You can use the data to support your discussion, okay? And yes, you're correct. These are very general statements, okay? Okay, what else is there? Well, I believe it lacks a bit of insight. It lacks insight, okay? And what else is there? Well, the language again. It keeps saying, we did this, we could try this, right? The modality is quite low as well. We could try using different plants and stuff like that. So it's not great. So once again, bad example, all right? Now have a look at this. Hopefully this is a bit better, right? Now, use paragraphing, okay? The experiment aimed to investigate the effect of different light conditions, natural sunlight, artificial light, complete darkness, on the growth of bean plants. The results supported the hypothesis, right? So look, you compare that to the hypothesis, right? That bean plants exposed to natural sunlight would exhibit the most significant growth compared to those under artificial light or in complete darkness. All right. Now, analysis of results. Remember, it happens here. So sunlight. Bean plants grown in natural sunlight showed the most robust growth with an average height of 14.6 centimeters after 2 weeks. See, they use specific numbers and durations. This result aligns with the expectation that sunlight, which provides a full spectrum of light, is most beneficial for photosynthesis. The healthy appearance of these plants with vibrant green leaves and strong stems further indicates optimal growth conditions. See, this is so much more detailed, right? So much better than before. Now, we're going to do an artificial light paragraph. Plants exposed to artificial light also grew, but to a lesser extent than those in sunlight. So it compares it, okay? And it also uses numbers again, averaging 11.6 centimeters in height. Although artificial light can support photosynthesis, it often lacks the full spectrum and intensity of natural sunlight, which may explain the reduced growth. See, you're trying to explain it. That happens in the discussion, right? Additionally, the slight variations in growth amongst the plants under artificial light suggest that factors such as light intensity and distance from the light source may influence the results. So it's pretty good, right? It tries to provide an explanation. Darkness, well, the plants kept in complete darkness exhibited minimal growth with an average height of only 4 centimeters. These plants showed poor signs of health, okay, such as yellowing leaves and weak elongated stems, a condition known as etiolation, okay, so where it doesn't get enough light, okay, it resulted in a very poor, appearance of the plant. The lack of light prevented these plants from performing photosynthesis, leading to stunted growth and poor health. This result clearly demonstrates the critical role of light in plant development. So that's the significance, okay? Now, see, you have further or future research and implications and limitations, right? So a lot of the times experiments have limitations, and it's quite useful and it's good practice to reflect on these limitations. The findings of this experiment underscore the importance of light quality and availability in promoting healthy plant growth for gardeners and agricultural practitioners. These results highlight the necessity of ensuring adequate light exposure for optimal plant development, okay? However, the experiment had some limitations. The artificial light setup might not have been optimal, as the type of light and its intensity could have been varied. Future experiments could benefit from using grow lights specifically designed for plants, which provide a more balanced light spectrum. Additionally, environmental factors such as temperature and humidity were not controlled and could have influenced the results. All right, and then obviously there's future research. But for time's purposes, I'll just leave it there, but it's just another thing that you can put inside your discussion. Okay. Now, conclusion. All right, we're nearly finished up with this. The conclusion is number 9. The purpose is to summarize the main findings. Okay, state whether your hypothesis or research question was supported. The tip is to keep it brief and concise. Reinforce the significance of your study, and then you can propose next steps or further questions, but that's optional. That's like a nice cherry on top, okay? Now, conclusion example. In this experiment, we found out that light affects how plants grow. The plants in sunlight grew the best, the ones under the lamp grew a little, and the ones in the dark hardly grew at all. This shows that plants need light to grow. We should always give plants enough light if we want them to grow well. More experiments could be done to see what else affects plant growth. Grace, what do you think about this?
It uses we. It uses the word. Well,
Yes, once again, it's
yeah.
we did this, we did that. It's not a passive voice, okay? It's less professional, less academic writing.
And it's also kind of simple.
Yes, it's quite simple as well, right? Look at this. This shows that plants need light to grow, right? It's quite simple. Okay, statements are very general, okay? It lacks the specifics once again, right? Remember, we need to summarize the main findings. So findings like the results, right? We should present some results here, okay? So let's have a look at this. This is an example of a conclusion. And as you guys would guess, this is the better one. It's a good example, okay? Now, the experiment demonstrated that different light conditions significantly impact the growth and health of bean plants. Plants exposed to natural sunlight exhibited the most robust growth with an average height of 14.6 over 2 weeks, indicating that natural light provides optimal conditions for bean plant development. In contrast, plants grown under artificial light, while showing some growth with an average height of 11.6, did not achieve the same level of development as those in sunlight, suggesting that artificial light is less effective. Plants kept in complete darkness showed minimal growth, averaging only 4 cm, and displayed signs of poor health, such as yellowing leaves, weak stems. These findings underscore the crucial role of light in promoting healthy plant growth, with natural sunlight being the most beneficial. Future studies could explore the effects of varying light intensities and durations to further understand the optimal conditions for plant growth. So this is future directions, and that's the extra cherry on top. Okay, so this is better. You can see they use more specific numbers. Okay, they describe the findings quite well, and they outline the significance and also, certain explanations here. So this is a good example. All right. Now, last bit, references. With all research papers, you need references. Okay, so the purpose is to list all the relevant sources used or cited in the report. Right, so for your background research, obviously you did research, right? So you'll have to cite the websites that you used or articles or books, stuff like that, okay? It allows others to locate the original materials that was relevant in writing the report. Now the tips, follow a consistent citation style. All right, maybe you guys have heard of Harvard referencing, Oxford referencing, but regardless of which one you use, make sure it's consistent. Don't use Harvard for one and then Oxford for the next, okay? Keep it all the same. Check what citation format is required by your school. All right, different schools often require different citation styles, okay? So just make sure you check with your teacher. And the last tip is the most important tip. I am guilty of this as well, okay? Organize and document references as you write your report. Okay, it's very easy for us to go click on the internet, be like, blah, 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 click, 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 do research, okay? And then once you write your report and you're finished and you're like, oh, I forgot which websites I used. Okay, I'm definitely guilty of that. So try to get in the habit of, organizing and documenting it as you do your research. Okay? Yes, Lucas, question?
Wouldn't you just go to history to check out which websites you use?
You could, but then what if you did your research over a month? All right, during that month, maybe I'm searching up recipes to cook dinner, okay, and places to go on my holiday. So it becomes quite difficult, okay? So if you do it in one day, yeah, fair enough. But a lot of the time, research takes much longer, okay? So let's move on to examples. Look, this example, not great, all right? They just got some links and they chucked it into the references and they're like, yep, good on you, done, references. That's not good, okay? This is a better example of a reference, okay? Now, these are all generated, I forgot which one I used, okay, but it's consistent, it's supposed to be consistent. And then a lot of the times, right, specifically for like Harvard referencing and stuff, right, we try to keep it in alphabetical order, okay, we try to keep it in alphabetical order. Sometimes it's the order in which the website or book is used in the report, but oftentimes it is alphabetical, okay? Now, I'm going to leave it there for now because we just went over all 10 of these, okay? It may be a bit long for you guys, so I'm going to give you guys maybe a 5-minute break to just get some water, refresh your brain, Okay, so we can talk about the last bit, which is a bit additional stuff. Okay, so go take a 5-minute break. We'll come back at 7:50 to finish off the rest of the seminar. All right. All right, sorry, I wasn't keeping an eye on the time because I was replying to some messages on the Zoom chat. But hopefully you guys are back at 7:51. All right, let's continue forward with maybe the last couple of slides. What are we up to? 52. So maybe like 18 more slides. Okay, so this is additional stuff. By this time, you guys would already know, the scientific method, the science report components, and how to write a pretty good science report. Okay, but now this is the additional stuff that I want to teach you guys. Okay, so let's have a look. First of all, I want to talk about variables. Variables. Okay, I've talked about this a little bit already. Okay, there are independent variables, dependent variables, and controlled variables. Okay, so these ones we've already mentioned. So independent variables are variables that are manipulated or changed by the researcher in an experiment, right? So remember, think back to your example. These are the light conditions, okay? Now, the dependent variable, it depends on the independent variable. Okay, it's a bit confusing, but this is the variable that is affected by the independent variable in an experiment. And in this case, remember in the last experiment, right? This is the plant growth. Plant growth. All right. So it is the variable that is measured or observed. So remember, we are measuring the plant growth. How high did the plant grow to? Okay, using a ruler. The independent variable causes the dependent variable to change. And of course, the different light conditions obviously affected the plant growth, as you guys saw. Okay? Now, controlled variables. This is what we haven't talked about. Okay? So, the controlled variables are variables that are kept constant during an experiment to ensure that they do not affect the outcome. Okay? So for example, when we talk about a last experiment, I only change light conditions, okay? And hoping that whatever happens to plant growth, it is due to the light conditions, okay? So I try to keep everything else controlled, okay? So for example, in this case, what is a controlled variable? It's the type of plant, okay? Remember, I used 12 bean plants. Okay, I do not use 2 bean plants, 5 sunflowers, 6 peonies, and things like that, right? That wouldn't be very good. Okay, what else is controlled? Well, it's water. Remember in the method, I said water each pot with 50 mils of water. Because what if I gave one of the plants too much water? Okay, what if I give another plant no water, right? That's obviously going to affect plant growth. Yeah, so water was controlled. Okay, so that's 2 examples. I'm sure there may be others if you think more deeply about it, but it is very important to have variables that are controlled. Otherwise, the plant growth will be affected by too many things, okay? And you won't know that it's because of the light conditions, okay? So it ensures that any change observed in the dependent variable is due to changes in the independent variable and not due to other factors, okay? Now, we talked about variables. I'm going to talk a little bit about accuracy, reliability, and validity. Now, this is the part where it gets a bit confusing, okay? So usually you learn these 3 things over the years when you go to high school, okay? But I'm going to try and introduce it to you now, okay? So question mark, question mark, question mark, because right now it may mean nothing to you. Now, accuracy, reliability, and validity are fundamental concepts in scientific research to ensure that results are credible and meaningful. Okay? Now, these concepts allow others to assess the quality and trustworthiness of the experimental data and conclusions. Okay? Now, if I did an experiment, Okay, let's say I did that same experiment I didn't give every plant the same amount of water. And like I said, if I use 2 bean plants, 5 sunflowers, 6 peonies, and something like that, someone's going to look at my science report and be like, that's not very trustworthy. Okay, it's not very good. Okay, so in that case, we have to assess accuracy, reliability, and validity. We have to make sure all of these are pretty good, okay, in our experiment. And then who remembers which part this one belongs in? Discussing accuracy, reliability, and validity. Which part did it belong in the science report? Anyone remember? So I'll go back up here. Which one did it belong in? Cynthia, yeah, unmuted.
I think it was
It's okay if you're not sure.
discussion.
Discussion. It is indeed discussion. Okay, so remember I said we'll talk about it later, and then here we are, we're talking about it. Okay, so let's have a look. Definition, start with definitions. Okay, accuracy. It refers to how close a measured value is to the true or theoretical value. Okay, how close is it to the theoretical value? Okay, so for example, all right, I'm measuring the temperature at which water boils. What is the true or theoretical value? What temperature does water boil? Tommy?
I think it's just 100 degrees.
Correct, 100, right? So it should be 100. But let's say I was doing an experiment and I measured it and it was like 130 degrees. Is that accurate? No, it's not very accurate because the measured value, which is 130, versus the theoretical value, which is 100 degrees, it clearly is very different. Okay, it's not good. It's not very accurate. So that's accuracy. How close a measured value is to the true or theoretical value. Okay. Now, because of this definition, accuracy, the only downside of accuracy is you can't assess accuracy if you don't have the theoretical value. You don't have any past data or anything like that, right? It becomes a bit more difficult in that sense, all right? Now, reliability, what does that mean? It refers to the consistency of a set of measurements or of an experiment. How consistent is it, okay? So, reliable, right? Let me have a think. Let's say you're measuring water again, the temperature at which water boils. You did it once and it says 100 degrees Celsius. You did it twice and it said 105 degrees Celsius. You did it 3 times and you said 130 degrees Celsius. Every time you do it, it's different. Is that reliable? No, because it's not consistent, right? So that's reliability. Okay, so let me get that off here. Okay, cool. Lastly, validity. Okay, so what does validity mean? It refers to the extent to which an experiment measures what it intended to measure. So for example, the aim, okay? So once again, if I'm measuring the temperature of the water, make sure it is the temperature of the water. Make sure you're not measuring anything else. Validity can be impacted by a lot of things, okay? And we'll quickly talk about it. So validity. When assessing validity, ask yourself these questions, all right? Is my experimental method and design suitable? Okay, is my experimental method and design suitable? So remember, right, why did I choose to use bean plants instead of other plants? Because it grows fairly quickly. Some plants take months to grow. I don't have months, so I chose the bean plant, right? Is my experiment testing what it's supposed to, right? Am I actually testing how light conditions affect plant growth, right? In order to ensure that, I have to have controlled variables, which lead to my next stop point. Does the procedure control all necessary variables except for the dependent and independent variables, right? Make sure you have controlled variables so that only the light conditions affect plant growth. Does your method include repetitions for a reliable result? Okay, do you repeat the experiment to make sure you're getting consistent data, right? So these are all things that you need to consider when thinking about validity. Okay, now I have a question for you. Take 20 seconds to think about this. Pretend you want to measure the temperature of the water which experimental setup is more valid and why? So to give you a bit of context, this is a can, this is a flame, you're warming up the can and the water, and this is a thermometer to measure the temperature. So which one is better, number 1 or number 2? Which one's more valid? All right, so Lucas, you got your hand up first.
Number 2 is more valid because one, the thermometer is held at a consistent distance, and also the burner is more consistent than the burner containing ethanol because it can flare up at random times, which would cause spikes.
Okay, so I mean that's pretty decent, but there was one other thing that I was thinking about. Izzy.
Something else that makes it more valid is it's labeled more.
Oh, not quite. So it's not quite the labeling that makes it more valid. So in this case, okay, let me ask Ethan. Ethan?
The number 2 is more valid because it is submerged in the water and it's not touching the container.
Good.
Because if you're touching if it's touching the container, then it means that it's more measuring the temperature of the container and the water, not just
Good.
the water.
Very good. That's it. So what Lucas said was good as well. I mean, I didn't make these diagrams. I copied it off from the internet. They should be essentially the same. Okay, but the biggest thing that is different is the thermometer. Look at this one. It's touching the container. It's touching the can. That means you're measuring the temperature of the can instead of the water. All right. Now, this one is submerging the water, like around the middle of it, right? So you're actually measuring the water's temperature, not the can. So this one is more valid. Okay? Because you're actually testing the temperature of the water, which is, possibly the aim of this experiment. Okay? Now, let's talk about accuracy versus reliability. People get it confused, and then I don't blame you. It is quite confusing. But look, I have 4 targets over here. Look at this. Okay, so obviously when you're shooting, in archery or whatnot, you want to hit the bullseye. You want to hit the middle. Okay, if you hit the middle, you are accurate. All right. Now, look at this. Is this accurate? Is this accurate? Is it hitting the bullseye? No, I see many shaking heads, so it's not very accurate. Now, is this reliable? Remember, reliability means consistency. Are you consistent? Not really. It looks like you shot there. It seems like it's just scattered around. So this one is poor accuracy and poor reliability, right? You're not hitting the bullseye, and then you're hitting everywhere else. Okay, kind of like randomly, it's scattered. Okay, look at this one. What do you think about accuracy? Is it good? Is it accurate? Is it hitting bullseye? Nope. But is it reliable? Oh yeah, you're reliably getting it wrong, right? So this one is poor accuracy, but good reliability, right? Okay, look at this one now. Is this okay accurate? I think it's okay. Okay, how about reliability? Is it reliable? It's like okay, but it's not great. Okay, so in this case, it's good accuracy, poor reliability. Okay, and this very last one, you guys will have guessed, it's good accuracy, good reliability. Okay, now, as you go through high school, you guys would, definitely come across accuracy, reliability. Whenever you think about it, come back to this diagram. Very, very useful diagram. Okay, I want you guys to think about this diagram years down the line. Okay, now, I want to highlight, look, a result can be reliable and inaccurate, right? Reliable, but inaccurate, like this guy, right? You're reliable, but is it accurate? No, you got it wrong all the time. It just happens it's consistent, okay? Now, a result can be unreliable and accurate. So in this case, yeah, you kind of got a few in the center, but it's not reliably there, okay? So this is something you should read and make sense of. And hopefully it does make sense from this example here. All right. Now, let's go talk about accuracy specifically, okay? So now that we understand the difference between accuracy, reliability, let's look at each one individually. So accuracy depends on, first of all, the validity, okay? It depends on the validity. For example, look at this, right? Remember this one, the thermometer on this one is measuring the temperature of the can instead of the water. So this is not valid. This is not very valid. And because you're measuring the temperature of the can instead, this is not going to be accurate, obviously. Not going to be accurate, right? So accuracy depends a little bit on validity, okay? Now, it also depends on the degree of errors, okay? So there's two types of errors, which I'll teach you. Systematic error, random error. Okay, I'll teach you this soon. But it also depends on sensitivity of equipment used. So for example, using a ruler that has only 1 centimeter intervals rather than 1 millimeter intervals. Okay, so usually when we measure something with a 30 centimeter ruler, there's millimeters, right? Imagine there's no millimeters. Okay, that's not very sensitive then. Okay, you don't know whether it's 12.4 or 12.5, right? That becomes more difficult, all right? Now, let's talk about the errors, okay? Because you guys may not have come across these two just yet. So systematic errors, what are they? So these are errors that are consistent and will occur again if the investigation is repeated in the same way. Usually a result of instruments that are not calibrated correctly or methods that are flawed. It usually has a consistent direction. Okay, so look, this is a very nice diagram. Okay, I stole it off the internet, but this orange line is your expected values. Now, systematic error looks like this. All the values are increased by the same amount. Can you see? It is consistent. Okay, it's in a consistent direction. It's all going up. Okay, it could all be going down as well, right? But this is systematic error, right? Look, it's all a bit elevated. Okay, another example, a scale that is improperly calibrated and also shows, and always shows a weight that is 5 kilograms more than the actual weight. Okay, so look at this. I drew a little diagram. This is a scale. Even when there's nothing on it, what is it pointing at? It's pointing at 5. Right? So this scale is not calibrated. So the next time I actually measure something which is 5 kilograms, what's it pointing at? 10. Right? So regardless of what you measure, it's always going to be 5 kilograms more. That's going to be a systematic error because it's always the same number off. Does that make sense? Yeah? Good. Now we talk about systematic error. Let's look at random error. As its name suggests, it's random. This guy's random. All right? It occurs in unpredictable manners, okay, and are generally very small, okay? Now, if a controlled variable is not kept constant throughout the experiment, this can cause random errors. So that's why you have to keep the controlled variables constant, all right? It can be minimized by repeating the experiment, removing outliers, and averaging, okay? So look at this. So this is my expected values, but then I get this random guy that's a bit off. See, this guy is called an outlier. So this is an outlier, and it is due to random error, possibly. Okay? Now, example of a random error. You guys definitely use a thermometer to measure yourself, okay, whenever you feel sick or have a fever, right? Surely. But then every time you stick it in your ear, it may read slightly different. It might be 37, 37.1, 37.2, okay? It's different every time because it depends on the position which you put it in, how deep you put it in, and blah, blah, blah. There's lots of things that influence it, right? But that's a random error, okay? So look, random error, example, you're measuring your temperature. It's 37, 37, a little bit higher, 37, 37, a little bit lower, 37 higher, 37 lower, right? It's random. It could go up and down. But what's the difference between this and systematic error? Well, look, this one goes up, it goes down, it can be up, it can be down. There's no direction, it's both ways. But for a systematic error, look at it. Systematic error is always in the same way. It's always in a consistent direction, okay? And then the thing is, look at this random error. This one's slightly higher, slightly lower, slightly higher, slightly lower. The thing is, if you take all of these and average it. If you average all of these numbers, this one and this one's going to cancel out. This one and this one might cancel out. So you might actually get something that's close to 37, right? So look at this. It can be minimized by repeating the experiment, removing outliers, and averaging. This is very important, okay? So do it a few times and average it. That helps you get a more accurate result, okay? Now, to improve accuracy, to summarize, you minimize the influence of random errors, all right? Try to minimize it, okay? You can't help it sometimes because it is after all random, all right? You remove the systematic error, right? So if it's the instrument that's not calibrated, just calibrate the instrument, okay? And try to use more sensitive equipment, right? Try to use a ruler that has millimeters instead of just centimeters, okay? Now, accuracy, a little bit more, okay? Now, the percentage error or the deviation can be used to evaluate accuracy, okay? You can evaluate accuracy using a formula. This is the formula that we use a lot of the times, okay? So percentage error is the experimental value minus the theoretical value, okay? You put in an absolute value, so it's always positive, and then you divide by the theoretical value, and you times it by 100%. This formula helps you determine accuracy, or evaluate it, sorry, okay? A percentage error less than 5% is typically acceptable. So it's typically all right if it's less than 5%. Anything more than 5%, not good, okay? Now, small example here. Try to use the formula. Do this example. I'll give you guys 20, 30 seconds. Give this a go. All right? It shouldn't be too hard, all right? But try it first. All right, some people are already getting it because it's not very difficult maths, right? Now, let me just write it out so everyone is on the same page. Percentage error equals to the absolute value of the experimental value. So what is the experimental value? Well, it's 106. I already know water does not boil at 106 degrees, but oh well, who cares? What is the theoretical value? What's the theoretical temperature water boils at? It's 100 degrees Celsius, right? And then you divide this by 100 degrees, should be 100 degrees Celsius. And then you times this by 100 because it's a percent. That's why you have to times by 100, okay? And then when you calculate this, it should just be 6%. And then because it's 6%, using my 5% rule, this is not really acceptable. Okay, you will see this formula as you go through high school as well, but that's just an example of how we use it. All right, that's accuracy now. Cool. Last but not least, reliability. Okay, now this is important because everyone, well, lots of students get this wrong, not everyone, okay? But reliability can be evaluated by repeating the experiment several times and comparing the results. Repeating the experiment does not improve reliability. Why? Does anyone want to tell me why? Okay, Tommy, you were first, so Tommy.
I basically think that maybe it's because like the more times you do it, the more likely it is to be different, and that's why it's harder to become like reliable.
Okay. I mean, that's a good thought, but that's not exactly why it doesn't improve reliability. That's a good thought though. Alice, what do you think?
I think if you like keep repeating it, but you get inconsistent results, that still doesn't improve reliability.
Exactly. I can repeat it 100 times and get 100 different results. Is that reliable? No. Did I improve reliability by repeating? Nope. Right? So what you can do is by repeating, you can evaluate it. You can think about whether it is reliable or whether it's not reliable, but never ever say, repeat the experiment to make it more reliable. Doesn't work that way. Okay? Yes, someone said, because you're doing the same thing wrong 100 times over. Correct, right? Now, the only way to improve reliability is to revise the procedure. So you can change the method a little bit. Okay, for example, you can use instruments that are less susceptible to random error, right? So random error, they're random, obviously, so they can make your results unreliable, right? So maybe you can use instruments that are less susceptible to that random error. Okay, and like I said here, reliability can be affected by random errors. So try to minimize that. Okay, now, this is very important. Repeating does not make the results reliable. Very, very important. Star, star, star. Okay, repeating allows you to evaluate whether the results are reliable. Okay, please, please, please remember this. Very important. Okay, now I have finished all my slides. Here are the objectives. I want you guys to read every objective with me now and see whether we met those objectives. Okay, look, understand the structure, key components of a science report. Okay, hopefully you guys understand the key components and structure. Okay, I'm going to assume that's a tick. Okay, learn effective writing techniques to enhance clarity and conciseness. Hopefully that's also a tick through all the examples I gave you. Hopefully you guys feel confident in your ability to write a high quality science report. Okay, hopefully that's also a tick. And the last bit which we talked about is differentiate accuracy, reliability, and validity. Hopefully that's also a tick. Okay, so there's only 4 objectives, right? Look at it and then try to tick them off. But hopefully I did a good job and then you guys actually met these objectives. Okay, now, like I promised, there's a question time now. You guys are free to ask questions that you may have, but please note there's tons of you, only one of me. We may not have time to answer everyone's questions. Okay? So, any questions whatsoever?
Is this like the last slide left?
Yes, this is the last slide left. So we saved a little bit of time to ask questions if you guys have any. Should we use subtitles in science reports? I would say, yeah, you are allowed to use subtitles, right? So remember when I wrote introduction, right? Introduction can be the header, okay? And then you can have subtitles, the aim, the hypothesis, and stuff like that. At what year level do you start writing scientific reports? I'm not too sure what year level that is, but in year 7 or even year 6, you begin to write very simplistic reports, okay? The structure I taught you today is sufficient all the way up to year 12, okay? And so that's it, right? So you may start from year 6, okay? And then it'll go all the way up to year 12. And even uni, if you decide to do science in uni, okay? I can clearly say for myself, I used it all the way up to uni and to this day, okay? Now, how long should a report be? Oh, reports may be different lengths, okay? Because you guys are younger, maybe it'll be like 400 words, 500 words in total, okay? But as you get older, towards university, it could be 2,000 words. It could be 3,000 words, all right? But your teachers will tell you how many words it should be, okay? Are we going to write scientific reports in selective? Oh, I don't know. I don't think it's in the selective test, but it's been a while since I've done the selective test. Okay, so I don't think so, but I'm not sure if anything's changed. All right. Did I actually do the bean experiment? No, I did not. All right, it took me a while to think of the hypothetical experiment to use, but eventually it came to me and then I decided to use the bean experiment. Okay. Is it required to write in full sentences or are dot points acceptable? Definitely use full sentences, okay? If you use dot points, it's not very academic, it's not very professional, okay? Can I please upload the slides? That's a good thing. Yes, I will try put it here. I'll try to put it inside the Zoom chat. It may take a while to upload though. Just give me a second. It may take a little while to upload, but I'm gonna put it inside the Zoom chat to everyone, okay? Which type of graph is used in a science report? You can use any sort of graph you want in a science report, as long as it's appropriately showing your results, okay? for year 7, will there be a test for this topic within Dr. Jim program? I'm not too sure. Okay, I'm not too sure, unfortunately, Michelle. Are we able to get the recording? I believe so. Okay, so we are recording this and then we'll be distributing it later on. So yes, there will be a recording. Would I use a paragraph inside the science report? Yeah, that could be possible. Okay, I could possibly use a paragraph. Okay, so you can use any graph you want as long as it's appropriately showing or displaying your data. Okay. So I have uploaded the slides to the Zoom chat. Hopefully you guys can see it from me. Okay. How do I do the good example for the references? Okay, so Sophie, there are online generators for references. Okay, there are online generators for references. So if you have the link of websites, links of the journals or textbooks that you use, you will be able to chuck them into the online website and then they will write the references for you. And then all you have to do is organize it, maybe in alphabetical order or the order in which you use it. Okay. Yeah, so if you just go online, like, free reference generator or something like that, you'll be able to find some. And there's all these different ones. They're able to do it for you. But just make sure you keep a list of the websites that you used, okay? Because otherwise, like I said, it's hard to go back and find them. Can scientific reports be a discovery? Yes, of course it can be a discovery. Okay, there's lots of things that we don't know about the world, okay, especially in the field of medicine as well, right? So yes, scientific reports can be a discovery. They can lead to new discoveries, they can lead to improvements, and yes, essentially the answer is yes. How should you adapt a science report for an invention? Oh, not quite sure for an invention. Well, it would be roughly the same, I would assume, even if you make an invention or come up with something completely new, because this invention has to come from somewhere, right? There must be some background research on it. Okay, it can't just appear out of thin air, right? So it should follow a similar structure, I would believe. Okay, can we make a report? Yes, you can, but it gets quite difficult if you make up the report. Okay, because you have to find the relevant websites, textbooks and whatnot. Okay. So someone asked a good question. In your discussion, is it better to include validity, accuracy, and reliability as an individual section or integrate it to the other parts? I would say just write a new paragraph and then comment on the validity, accuracy, and reliability. Okay, you can write 3 different paragraphs for it as well. It doesn't matter, right? You don't have to write one big chunk. Okay, you can use paragraphing as you would think is appropriate. What is my top tip for writing a report for a test without preparation? My top tip is always plan, okay, plan it. So if they give you a hypothetical experiment, plan it. Pretend you're the experimenter and you're doing this experiment, right? So you have to pretend you're doing it. And imagine all the little steps that you're going to do and write them out, convey them properly to, well, the marker or whoever's assessing you, okay? Why should we learn to write a scientific report? Well, in the first few slides, I've mentioned, it teaches you lots of skills. Okay, it teaches you how to communicate properly and effectively. Okay, it teaches you critical thinking, teaches you data analysis, and lots of things. Okay. Okay. Any other questions? Should I choose biology, chemistry, or physics? I'm not too sure for how to answer that. They're all quite different, but in the end, they are all branches of science. I personally like chemistry the most. It just makes sense to me. Okay, some people love biology, some people love physics, but it really comes down to your personal preference. Okay, physics was a bit difficult for me because there was a lot of abstract concepts, things that you can't see. Okay, So I found that a bit easier with chemistry, but I mean, it's down to personal preference. Okay. Do all science reports have the same structure? Yeah, I believe at least most of the science reports that you'll read as you get older will have the same structure. Okay. Yep, the recording of the meeting will be distributed, I believe, by Dr. Jin and Cogito Tutoring later on. So maybe just wait on that. Okay, but yes, we are recording it. Any other questions? How to fix outliers if there is one? Well, you don't really fix outliers actually. You actually should identify the outlier. Okay, in your report state that, you know, this value is an outlier. Okay, so I'm going to exclude it from my data analysis. So you don't really go and fix it. It's important that you don't go fix it because otherwise it seems like you're manipulating your results, and that's not a good thing. Okay, so in science, we don't want to manipulate the results. All right, we want to keep it honest, okay, as they happen. All right. Okay, cool. So, I mean, there's still some questions, but unfortunately, you know, there's quite a few of you, only one of me. So I may slowly try to conclude the seminar, okay? If there's no other questions, maybe I'll take one or two more if you guys have any more. But otherwise, hopefully you guys learned something, okay? Take away something from the seminar and continue and bring that, you know, into your future studies. All right. Okay. Is scientific reports useful for our everyday lives? Not super to our everyday lives, but for your academic life in high school, yeah, I think it will be useful. Okay, it helps you get better results if you understand it better. Okay. All right. Cool. You guys are very welcome. Okay, you guys been great as well. So thank you very much. Hopefully see you guys one day, one of these days in another lesson or something. Okay. Cool. You guys are free to leave. See you guys.
Thank you, Kevin.
Thank
No
you.
problem.
Thank you.
Thank you.
No
Bye.
Thank you.
Thank you, Kevin.
problem. See you
You did a
guys.
great job.
Thank you.
Thank you.
Thank you, Dr. Jin. Thank you.
Thank you.